Don't pretend they're not here.
Okay.
It's just you and me having a conversation.
Okay,
Private
hi.
conversation.
Okay, I'm
Okay.
Paulina. I'm doing my PhD here in Nottingham. I'm in my 4th year, so I'm writing up my thesis.
Oh, poor you.
Yeah.
That was the worst time of my life, actually.
It is.
Because it's so
writing...
lonely, you know, it's just you and the word processor.
But it's not too bad. I come back to campus every now and then, so I get to see like my friends and all of that.
Right,
So,
okay.
so it's been lonely, but at the same time, it's been nice to have people around who understand the whole process of getting a PhD.
Yeah.
Yeah. So actually today, I think like I mentioned earlier, we all know the discoveries that you've made and what you've been awarded for your Nobel Prize. So I just wanted to ask you a few questions about your journey in making those discoveries.
Sure.
And to inspire some of us young scientists, especially for us coming from a country where science and research is not exactly at its
There's not such a long tradition. Yeah.
Yeah, so actually I wanted to ask you about your motivation in pursuing your career. So what I did is that I watched your Nobel lecture.
Oh, really?
Just parts of it.
I think I've never watched that.
Yeah, really?
It's too embarrassing.
No, I thought it was okay. Yeah, so I think you mentioned something, and I just want to quote you on this, and you said, I didn't particularly like biology at that time. I think this was when you were in high school, but you said you were very good at it, and hence you kind of pursued it. So I wanted to ask you what motivated you to pursue science at a young age, even though it wasn't exactly your passion at that time?
Well, it sort of was, but I would really have loved to have been a physicist.
Oh, really? Okay.
And what I liked doing as a little boy was to take old radio sets apart or build new. You find a lot of my generation have the same thing. It was after I was actually born during the war, and sort of grew up after the war. And the Tottenham Court Road, where I now live very close to, I never dreamt that I would achieve this. It was full of war surplus components, so you could easily buy these components and put things together. But mostly what I did was just to find old radio sets and take them apart and think how beautiful they were. Nowadays there's nothing you can take apart. I mean, you can't even open a mobile phone, right?
True,
and if you did, all you'd find was integrated circuits, and you wouldn't know what the hell they were. So I don't know, that sort of route to entry seems to be blocked. But we also did some other things, like we discovered a lead mine in the garden. At the back of the house, there was a sort of deserted lot which had been, I think, 4 little tiny cottages, and which had been demolished and taken away. But the lead piping, which provided their water, was still there. And so we found this vein of lead, and we would cut it up, we'd dig it out, cut it up, and then melt it down in a blast furnace, which we constructed in the back garden.
You managed to melt the lead?
Yeah.
Wow,
we sort of put it in a coffee can tin, Nescafe tins, with I've coded how they were propped up. So there was a lot of that kind of thing. Or making explosives. I learned that if you mixed fertilizer with sugar, it would explode, and then we had to find a way of igniting it. And it turned out that we had an electric train set, and that had a transformer. And if you put steel wool between, it burst into flame, and that would ignite the explosive. So we sort of made explosions and things like that. So these, it was things like that, I think, that really made me scientific. And then I had 2 or 3 really excellent science teachers. And the one who made me be a biologist was a guy called Gert Sommerhoff. And how I learned that I was good at biology was because he set the same exam to the whole school from ages 6 to 13 or something like that, okay? And I was in the middle of the school, 400 boys, I don't know, 450, something like that. And I came out, I was 13th, I think. Not top of the class, but did terribly well. I never did so well in the physics exam, but this was a biology test. And so I realized that I was terribly good, just naturally. I hadn't revised or anything. It was just that my mind sort of worked in a biological kind of way. And I think it did actually, it sort of does naturally. I mean, I'm very lucky in that respect. So after that, I was maybe 12 years old, and or 11, I'm not sure. I never really had to take any career decisions, which was great, because I mean, I did worry about whether I would pass the next exam, because of course you have to keep doing that. And you know, I was constantly, I mean, you know, they thought that I ought to get a scholarship to Cambridge, and I didn't, but I scraped in somehow. I mean, one of the problems was that I had to take a botany exam, and I hated botany.
Oh, me too. I don't
I was no good at it in those days. Now I actually love plants, and I love coming to Malaysia because of the jungle and the incredible, beautiful variety of plants that there are here. And some of those they grow indoors at home, you know, and that kind of thing. And then I had a really good secondary school teacher who was a zoology teacher. And one of the amazing things about a lot of my teachers, they almost all had PhDs themselves, so they really knew what they were talking about.
So
I was very lucky in school. This first guy was he was a German refugee, actually, Jewish German refugee, and he had a PhD in something.
Was that common at that time for teachers?
No, I think that was really rather unusual. I mean, it was quite brave of a headmaster to hire a German, because, you know, the Germans were our enemy during the war. But he was terrific about explaining complicated things in a simple way. And he had a science club and things like that, you know, and made radio-controlled things. I was never up to that. I didn't understand how that worked. It was a pity. And I had a friend, a very close friend of mine. We used to go on long cycling trips together around and about, and then later on abroad. He was a physicist, and actually turned out to be a very, very good physicist. So I sort of compared myself to him, and I realized at once that I was just not nearly so good as Peter. And so, you know, there was no point in worrying about that, because it just didn't come naturally to me. I mean, I always thought about things the wrong kind of way. But in biology, it was the other way around. And I sort of knew and what else did we, you know, we used to electrolyze solutions of salt and make chlorine and hydrogen gas, you know, and then smell them and sniff them. And in chemistry it was fun, because you had to identify chemicals, and you burnt them and you sniffed them and you did various tests and you find out what they were. So and I, you know, just it was good fun. But then I really sort of got into my stride, I would say, as a university student in Cambridge, and there it was much more serious.
So how did you find your direction in research after that? So you went into, you know, undergraduate?
Well, you know, it was personal, really. I was, you have to understand that messenger RNA was discovered while I was an undergraduate. In fact, I remember my friend Bridget coming into a lab and sort of waving a copy of Nature, say, Tim, they've discovered messenger RNA. It was fantastic.
Times are different now.
And so that was all in the air, and so we were right at the frontiers. In our lectures, they would explain the latest experiment, which had just been published. It was super exciting. And there was a time when Jacques Monod came and gave 2 lectures in the big chemistry lecture theatre, and these were absolutely marvelous. I mean, gosh, what a revelation, how genes are controlled in bacteria, you know. And I went to my supervisor, my undergraduate supervisor, and said, you know, is this true? And he said, well, it's too early to tell, because he hadn't been reading the French literature, because it was in French.
Okay. So it wasn't translated.
But the other people, well, a lot of it wasn't, but actually there was a famous review which was published in 1961, I think, which laid it all out, and it was so beautiful. And, you know, and then we had lectures from people like Max Perutz and Fred Sanger and Sidney Brenner. So these were the people who were actually making the, you know, huge strides at the time. You know, Sidney was the person who discovered messenger RNA, and we had lectures from these people. Yes. So this was very, you know, and you sort of realize from listening to these people that, at least it probably took a little while later on, I sort of realized, you know, actually, you know, they are giants, but on the other hand, you know, they're only mortal. I mean, in some respects, I found I knew more than they did. And that was sort of encouraging eventually, because it was, you know, yeah. So you sort of begin to be feeling that you're putting your feet forward and there's landing on solid ground, and then you have enough confidence to put the next. So it built up, I think, gradually. I mean, to begin with, I had absolutely no idea what was going on. I was fantastically ignorant and stupid. I made every single mistake that you could possibly make. And I think I was pretty good at not repeating any of those mistakes. But you have to make an awful lot of mistakes to get good at something, in my experience. And so, you know, that was how it worked.
I want to talk a bit more about making mistakes. I think personally, I myself have made a lot of mistakes, and I think failure is a very, very big part of research. You know, you do an experiment and it fails. And
it mostly fails,
yeah,
right?
it mostly fails.
Partly because your ideas are wrong, and partly because you're incompetent. Or the machine isn't working properly, or all of those things. And it's can be quite difficult to troubleshoot sometimes and decide where the problem lies.
Yeah. So I think something that you mentioned in the lecture as well was that you mentioned that your lab burned down.
Yeah.
Yeah. So I guess just to link it back to failures and things like that, I wanted to ask you what were the low points in your career, and how did you overcome, you know, all of these failures and mistakes, and how do you, you know, just overcome that?
Well, I think the answer to that is it happens very often, and, you know, setbacks are much more common than triumphs. I mean, I think real triumphs come every few years, actually. But you have little micro triumphs, sort of, when I was sort of making a lot of constructs and things, which I had spent, that was sort of typical. You know, you did a transformation, and hopefully there would be no clones on the negative control and lots of clones on the positive control, and then you got one or two in, you know, and then it was always fun coming in the morning to see whether everything had worked according to plan,
definitely.
you know. So, and these were usually trivial, minor steps in a grand pathway, but it's sort of doing that is good. So you've always had things that sort of always worked, which sort of reassured you that you hadn't completely lost your marbles. And then there were the real experiments where you really didn't know what the answer was going to be, and, you know, you had to interpret it as best you could. And sometimes you made progress, and sometimes you found you'd guessed wrong, and you had to go back to square one. And the thing is, there are lots of different kinds of projects. There are some where you just sort of crank the handle and it's going to work, and you're not going to set the world on fire, but it's kind of satisfying because you sort of find things out, and that's good. And maybe you sort of get a little more skillful in doing experiments as a result of that. And then there are the times when you really realize what's going on and make a discovery, and that is really wonderful. I mean, you know, it was a real high. It's sort of, you know, it's the most amazing feeling when you get to the bottom of things.
And when you talked about doing an experiment, and sometimes we get stuck and we have to go back to square one, sometimes I feel like there are times where I don't know where to go, and you know, we kind of face like that. It's kind of like a writer's block where you just don't know where to go. So how do you get out of that?
Well, you just have to keep
digging.
Keep going, actually. It's just very simple. You just have to keep going. You have to be, you know, if you get blocked and you want to find something out and you keep on pushing it, you know, one thing that's very important is maybe there's a secret back way round and you could attack the problem from a different, or maybe the problem is just too hard for you and you should give up. That's pretty difficult to judge, actually. And it depends on, you know, every single little project has its own problems. I think in many ways the worst time is when you've just made a great discovery and you know, you've figured out what's going on, then knowing what to do next is awful, actually, because you sort of know what the triumph feels like, but you sort of fall into this bottomless pit of unknown, because you just don't, you know, you've solved one problem, so what are you going to work on next? And you've no idea. So I was very lucky that in the end I always sort of came back to something, but in one case, you know, it took more than 10 years. And it gets to you, actually, that, you know, you start. That's why people sort of give up and start teaching on things, I mean, which is perfectly good. And I think teaching runs strongly in the family. My brother was a teacher, my father was partly a teacher, an uncle was a great teacher, and so on and so forth. So explaining things and bringing on the younger generation was always important to me. But I preferred to sort of teach in the lab, you know, not try to lecture to people, but actually show people how to do experiments and how to interpret them, and how one experiment led to the next experiment, led to the next experiment. And that was, because of what we were doing at the time, that was really good, because you could do several experiments in a day. So it really, you know, it was lovely.
Okay. You talked about making, you personally, you've made a lot of new discoveries, and I think at that time when you discovered cyclin, it was kind of like a disappearing protein, and these were things that didn't happen at that time. I was wondering, how did the scientific community respond to that? You know, it's something that, you know, has...
Well, with a complete silence, actually, because it was so unexpected that they didn't know what to make of it, which was very, very lucky for us. So that paper, I made the experiment on I saw the result of the experiment on July the 22nd, 1982. And I know that because it's written in ink, radioactive ink on the autoradiogram. Okay. The paper was published the following year. Yeah. And amusingly enough, when I submitted the paper to Cell, and the reviewers came back saying, "Dear Tim, we will take your paper, but in nothing like its present form." And one of the reviewers was really hostile, you know, and he said, "This is rubbish, complete nonsense, can't possibly be." And and my mind was all on a completely different topic, which was translational control of messenger RNA translation in sea urchin eggs at the time. So I didn't really, and I didn't know anything about cell cycle control, so it was all sort of there was nothing really to sort of get your teeth into. It was very hard to write the paper. But I revised it, you know, and the other funny thing was you couldn't do any more experiments, because after July, the end of July, there was no more sea urchin eggs, so not until the following July could you do any experiments. So that was also a bit, but I was so elated by the thing, and I told everybody I could possibly find, because I was very proud of this discovery, and they all thought I was crazy, you know. And it was very funny that, I mean, it must have been that I sort of knew enough about the history and understanding of cell cycle control that I realized that this was a really, really important principle, and that I'd sort of unlocked a key which other people didn't know about. And it was clear that most people just didn't know enough about it to realize the importance of the experiment. So it was their ignorance rather than my bravado or something, you know. So I was very confident that you know, it was really, really important to find out what this thing was. And the problem was that it was a sort of backwards problem. In the previous problems, you know, I had an effect and I needed to find the molecular basis. It's much harder when you've got a molecule and you've no idea what it is, because actually it's really, really difficult to, you know, even in the human genome contains 30% of genes that we have no idea what they do. Gradually that number is reducing, actually. But working backwards, it was really troublesome. And we were stupid, and really, actually, in retrospect, we were slow. When I say we, I mean, it wasn't just me. I mean, I had a lot of very smart friends, really good, you know, brilliant people, much cleverer than me, and we were all equally blinded by prejudice. And actually, the simple prejudice was that there was a protein kinase known, a very commonly found protein kinase called protein kinase A, which is stimulated by cyclic AMP. And cyclic AMP has an inhibitory subunit. And the way the cyclic AMP turns it on is by removing the inhibitor. Nobody had ever described a protein kinase which required an activator. And because of that, we never even, that idea, a very simple idea, and theoretically nothing against that idea, but it just never ever crossed any of our minds until we were finally forced upon it by the structure of the thing. For the active thing had these two subunits. One subunit was CDC2, the other subunit was cyclin B. And, you know, so why did it take sort of 4 years or 5 years to test that? It was so obvious in retrospect. So how could it have been so non-obvious when you didn't know what? That's what I find fascinating about these kind of things. And I think there's an awful lot of biological things which are probably like that. You have prejudices which you don't really properly interrogate yourself.
Yeah, definitely. I think I agree. Because for us, we were taught to, learn and memorize the fundamentals. But how do we deal with the need to, we have to learn, we have to memorize whatever.
Yeah, and where do you draw the line between questioning
yeah.
you? So, I mean, we were taught as undergraduates not to believe a word we read.
Oh, okay.
Right? I mean, we were actually said that, especially in reviews and textbooks, because they were sort of simplifications of the real thing. Right. So always, and we were also taught very much so, when I asked people to write essays on things, which I had to do for years and years and years of my life, I was always writing in the confidence, in the margins, what's the evidence for this? What is the evidence? What is the evidence? How do you know that? So that was the way we were taught, not how does it work, but how do we know how it works, and how would you find out, how would you take the next step to push the frontiers of knowledge forward? So I think that was very helpful and probably somewhat unusual.
Yeah.
I mean, when I was shocked by my younger daughter's biology textbook, because it had things like memorize the 5 adaptations of the pine tree.
That was what we were taught. So we just have to,
And it didn't actually explain what the adaptations were, it just had a list of things, It didn't tell you how people knew what, it was awful. And, Dad, don't explain, just what is the answer, So this is very interesting. So it's complete misconception of what the scientific process is like. And it's so much more interesting to find out for yourself and to sort of wonder about things. And we were taught silly things like plant sex, for example, is really complicated. The triploid endosperm and how pine trees work. I still never really figured that out. So why do they teach that stuff in school? They don't really sort of teach you. And I never really, until I saw it with my own eyes, understood the difference between mitosis and meiosis. I mean, these are very simple but sort of deep concepts that need proper explanation, and you need to be sort of really, really shown how it works. And it's not easy to understand some of them, and just reducing them to lists of memory is pointless, in my view. Absolutely pointless. And boring and
just to wrap up everything, I guess the last question is, I do think that in your opinion, you don't agree with the way that science is taught at the moment. So what would be your input as a scientist in how science is being taught to us at the moment, and what do you think could be improved upon?
I don't know. Because there was a thing called Nuffield science in England where everything was done by experiment. And I don't think it worked all that well because people didn't really understand the nature of the experiments they were doing. There were some very clever ideas, but you've got to sort of find out for yourself. You've got to walk into a field and say, why are the nettles growing over there and not here? And then you've got to do some experiments to test your ideas. And that you can't do that with a class of 30. You could do with a class of 2 or 3. So I think there's a big problem there. And I don't know what the solution is. I think, you just have to let people get on with it and hope that they like it. And getting down to basics, it's sort of hard, actually, really hard. I do think science is a hard life, actually. It's not, it's true, it's really, really difficult. I mean, the idea that anybody can be a scientist is nonsense, actually, I'm afraid. I mean, it's like saying anybody can be a concert pianist. That's obviously nonsense. Anybody can be a Premier League footballer. Obviously nonsense. But somehow science is open to everybody. It's just not true. You need but having said that, there are an awful lot more different ways of being a good scientist probably than there are of being a good footballer or a good concert pianist. So actually, it's much more open. But you have to find your own way. And I don't know what the perfect way of introducing. You have to stumble on it. You have to find it for yourself. It's really something that bubbles up from inside you. don't you think?
Yeah, I agree. There's no system that fits everyone. You just
Absolutely,
have to
we're all too
discover on
different
your
from
own.
one another.
Yeah.
Yeah.
Yeah, it's been lovely talking
Well,
to
it's
you.
been great talking with you too. Thank you
Yeah.
for such interesting questions and
Thank
being
you.
sort of a
Yeah,
sympathetic audience. I'm sorry I talk so
Oh
much.
no,
you know.
it's fine. It's a fresh perspective. I've learned a lot from you.
Well, you too, and I hope you. As I say, writing the thesis was just the worst time of my life.
I hope it's
Just-
over soon.
Yeah, no, and we want to get back to the lab or whatever it is you want to do next.